Good evening, everyone. It's a pleasure to be with you. I wanted to provide a little additional information on myself before we start. So I've been with P&G for 35 years in R&D. I got my postgraduate degree in physical chemistry. I was a solution physical chemist, but after I came to P&G, I became somewhat of an oral biologist. I've been doing paste, rinse, and brush and whitening bleach research for that time. I've been doing gum health research since the late 1980s, about 28 years. So I was one of the developers of our Pro Health paste and rinse and our 3D whitening paste. And I also was involved in the development of the high bioavailable Stannous fluoride systems that we use in our Pro Health toothpaste around the world. So that's a little bit about me. So I want to talk to you this evening a little bit about the advantages of using antimicrobial toothpaste. So I'll start out by saying one of the surprises in my career has been that myself and my colleagues that I compete with at other companies, we developed sets of products in the 1990s and 2000s that had antimicrobials included in them to provide improved gum health to patients. And myself and my colleagues that work at other firms are always surprised that these toothpastes have about a 25% share of the market, and we're even more surprised that your average dentist doesn't always recommend them, and considering the amount of research that we've done on these. And so I want to talk to you a little bit about their advantages and make the case for recommending these for all of our patients. So today I want to talk about what we've learned about our Stannous fluoride toothpaste in two specific things: plaque toxicity and how to measure it, and how Stannous fluoride toothpaste actually reduce plaque toxicity. First, I'd like to thank the researchers that have been involved in the research I'll present today. You can see a list of them here. There's many, many, many people in the background doing this work, so I want to make sure I give full credit to all the people that made the research you're going to see come to happen. So how did all this get started? Well, as I told you, I had done research for a number of years on toothbrushes and toothpaste and so on, including antimicrobial toothpaste like Stannous fluoride and so on. And one of the things we noticed in our research was that it's true that if you control dental plaque, you tend to improve gum health, but we realized that not all plaque control provides the same level of gum health benefits. And I'll show you an example from our Stannous fluoride system to kind of show you how this is so. So this is from a published clinical study from Gerlach and Amini in 2012. You guys can look it up if you want to. Stannous fluoride clinical effects on gingivitis here were substantial. So after 3 months, they measured 74% versus a placebo paste control. So that's a pretty substantial improvement in gum health. And yet when we look at the plaque on the people's teeth, we only see about a 25% reduction in their dental plaque. So the question then becomes, how does a 25% reduction in dental plaque, and you can see the plaque reduction here on the placebo paste was Crest Regular and the test paste was the Pro Health toothpaste. How does that 25% reduction in dental plaque actually translate into the kind of gingivitis benefits that we see? So in the clinical study on gingivitis, we saw 74% reduction in gingivitis, but we only saw a 25% reduction in dental plaque. And so this was a surprise to us, and the gingivitis benefits were much greater than we would predict from simple reductions in plaque quantity. So honestly, in our laboratory, we asked ourselves, what the devil is going on? How do these antimicrobial toothpastes reduce the gum disease? And by the way, we see this with all of the antimicrobial therapies that we put into mouth rinses and toothpaste. So this is a common observation. So it was decided by us to focus our research on plaque toxicity, and we hypothesized that therapies, any therapies, which reduce gum disease must show effects on plaque toxicity. And we wondered whether modern techniques to measure microbial toxicity might show the benefits, including those for Stannous fluoride. So some background before we start with the evidence. So the reason we wondered about plaque toxicity is in the following sets of slides will describe this for you. Where gum disease actually happens is in the sulcus, as all of you know, and dental plaque forms both above and below the gum line. It's the plaque in the sulcus, below the gum line, that's actually causing the gum disease, the gingivitis and the periodontal disease. The plaque bacterial composition varies a little bit with location. So above the gum line, you see the green cells up there. Those are gram-positive bacteria. Below the gum line, you have more of the black-colored images there. Those are the gram-negative anaerobic bacteria, and they predominate in the sulcus where the oxygen is very, very low. But why does the sulcus plaque cause inflammation? Why do these gram-negative bacteria create the redness and bleeding and the inflammation that we see when we get gingivitis? Well, believe it or not, the clue to the discovery of this starts out in the 1990s with fruit flies. And so it was in the 1990s that Christian Nusslein-Volhard and Klaus Wieschaus did work on fruit flies and identified a set of receptor molecules that were responsible for changes in the growth of the actual larvae. And it's Beutler, Medsitoff, and Janeway at Yale. Beutler was in Texas. They did studies to show the same receptors were present in mammalian cells. And these receptors that they identified, these genes that they identified, were responsible for the body's response to infectious diseases, including the innate immune system and the activation of the adaptive immunity, including antibodies. So there were these resulted in 2 Nobel Prizes. There was a Nobel Prize in '95 and then 2011 for these discoveries. 2 things came out of these discoveries. The first was that the tissues of the body contain receptors which act as sentries for the identification of infectious diseases. These receptors are called pattern recognition receptors. One type of these receptors is called toll-like receptors. You're going to hear about that more this evening. The second thing that they discovered was infectious pathogens, viruses, bacteria that cause diseases invariably contain molecular patterns in their composition which are unique and different from the cells that we have, the host cells in our bodies. These molecular patterns are called pathogen-associated molecular patterns. And it turned out that these toll receptors were evolutionarily conserved across all the species that we see throughout evolution. So this explains how the innate immune system actually evolved during the evolution process. The human beings contain an adaptive immune system too, with antibodies, which is more sophisticated than what some other invertebrates have. So here's what we know today. We know today that the gingival tissues, and there have been lots of studies to show this, contain receptors throughout which act to survey for the presence of bacterial pathogens, including those which are in our microbial plaque. So the host cells in the gingiva that contain these TLRs are epithelia, endothelia, gingival fibroblasts, periodontal ligament fibroblasts, and all of the various inflammatory cells that get recruited to fight the infection, including the leukocytes. Now, what happens when the host cells contain these receptors is that these receptors are located on the cell walls of the host cells. So they're located on the interior of the gingiva, the sulcus. And when the bacteria interact with the toll receptors on these cells, they send signals to the nucleus of these cells, and that activates the inflammatory process. It provokes the immune response. That in turn results in cytokine expression and all of the symptoms of inflammation that we see, including redness and swelling and so on and so forth. To make matters worse, the pathogens below the gum line, these anaerobic bacteria, they also have metabolic activities, which further act to increase inflammation. So they produce a variety of species, and the 4 main groups are shown here: short-chain fatty acids, volatile sulfur species, amines and amides, and enzymes like gingipanes. And all of these metabolites act to increase inflammation. In many cases, they increase the reactivity of the toll receptors with the actual endotoxin or other activating components of the microbial plaque. In some cases, they increase permeability of the tissues so that more of the tissues react. So toxicity of microbial films can be defined as molecular patterns on the bacteria. That includes two components, LPS, lipopolysaccharide, it's also called endotoxin, and LTA, which is called lipoteichoic acid. Those are the two molecular patterns from the bacteria that we have in our mouths that actually can activate the inflammatory response. And then there are also, secondly, the metabolites which are produced by those bacteria, again, in the sulcus. So a more complete picture of why we get gingivitis and why we get periodontitis is that the dental plaque below the gum line are producing these toxic species, the LPS, the LTA, the short-chain fatty acids, the volatile sulfur components, the enzymes, the amines. And these are diffusing and interacting with the tissues, which are sending signals to activate the innate immune response, and that activates the inflammation that we see. So you can see that in this diagram, the recruitment of all the leukocytes to fight the infection at a site of chronic gingivitis. So on this basis, we knew that that was what the state of the science was up to 2010 or so. So then we started to ask ourselves some questions. Can we measure toxicity of plaque in the laboratory? And this is very, very contemporary, measuring the actual toxicity. Lots of researchers have counted numbers of bacteria, but not all bacterial species produce the same number of metabolites. Not all bacterial species have the same amount of LPS on their cell membranes. And so toxicity isn't the same as number of bacteria. So we asked ourselves, can we measure toxicity of plaque in the laboratory? Do therapies including Stannous fluoride influence toxicity, and do these effects occur clinically? So in order to do this, we needed to develop and apply some new tools. And so for just a couple moments, if you'll let me, I want to talk about some of the techniques we use to evaluate toxicity of the pathogens. There are 4 of them: dye binding studies, reporter cell assays, cytokine expression assays, and a tool called Metabolomics. And I'll talk just a little bit about each of these before we go into the data on Stannous fluoride toothpaste. In the dye binding studies, it turns out that detecting quantities of LPS and LTA are extremely important, because if you're making medical devices and you sterilize them, you may kill the bacteria, but you may leave the LPS on the suture. You may leave the LPS on a piece of equipment, and that can still act as a pyrogen to induce a fever and an inflammatory reaction in people. So you need to make sure that when you sterilize these instruments, you have to get all of the endotoxin and so on off of the surface. So they developed dye techniques. There are 2 of them. There's a Bodipi cadaverin assay, they call it, and then a second assay they call LAL. And in both of these assays, if you have LPS or LTA, which is on the left-hand side of this slide, and you react it with a dye molecule, it interacts and it changes a fluorescence signal, and that can tell you whether LPS or LTA is in solution. So it's a very, very simple assay that even a high school student can run. A second type of methodology we can use to study plaque toxicity is a much more sophisticated one. These are reporter cell assays. So living cell lines can now be created by molecular biologists, and we use cell lines that have been created out in California at Invivogen. And they contain active toll receptors that have been constructed into their cell membranes. And those toll receptors are in turn linked to enzyme systems inside the nucleus of the cell lines, which allow the cells, when the toll receptor is activated, to create an enzyme change, which we can actually measure in solution as a fluorescence. So what this allows us to do is it actually allows us to confirm if ingredients or formulations can interfere with the toxic reactivity with the cells. We can do it in vitro, and we also, I'll show you later, we did this with in vivo samples as well. So these cell lines have been specifically designed for researchers all throughout the world to be able to study antigen reactivity with cells, and that's how we use them in our laboratory as well. So this is what the reporter gene assay looks like. You take bacteria, or you take the actual toxic parts of the bacteria, you interact them with these reporter cells, you incubate them for a period of time, and then they give a luminescent signal, which you can actually read, and it's proportional to the amount of cell activation. Another method to look at plaque toxicity is to actually look at cytokine expression. These assays are a little bit more difficult because they require the use of a number of reagent conditions to measure. But you take human monocytes human monocyte cultures and you interact them with the toxin and you incubate them, and then you can actually measure the secretion of cytokines into the media. And we use this assay to reinforce actually the validity of the cell reporter models, because this assay is too complex to run hundreds and hundreds of samples with. And we use this to measure toxicity of subgingival plaque samples in limited circumstances. So in the cytokine assay, the subgingival plaque, or the LPS or the LTA, is mixed with the monocyte and it's incubated, and then you measure the cytokines using detection kits. The last methodology I wanted to talk about is a systems biology technique called Metabolomics, and here we're analyzing the whole saliva. So this method permits us to do a chemical analysis of all the metabolites, the species being generated in your oral cavity and by oral bacteria in the host. So we collect saliva in the morning, we stabilize it, we carry out an analytical measure, and we can measure various metabolites. And several of them are key to activating gingivitis and inflammatory processes, and that includes propionic and butyric acid. So those are some of the techniques we use to evaluate toxicity of plaque and bacterial pathogens. So I'll walk you through some of the data we have in our laboratory on Stannous fluoride toothpaste, where we've used some of these methodologies. First, the laboratory studies. So the focus of these studies was to confirm and establish the methodologies to test for microbial pathogenicity and their effects on inflammation pathways. And we used these to see whether Stannous was able to actually bind the antigens in the bacteria and render them less active in promoting gene expression associated with the toll receptor activity. So we were actually looking to see if Stannous fluoride could detoxify the LPS or LTA toxins on the bacteria. So these are the results of the dye assay. So let me orient you to the slide. On the y-axis, we're measuring the percent blocking of the LPS with the dye. On the x-axis, we're looking at the concentration of Stannous in solution, Stannous fluoride or Stannous chloride. And as the concentration of Stannous is increased, you can see that the blocking starts to occur after 1250 micromole. And then once we get up to a higher concentration, you get increased blockage of the actual dye with LPS. You'll notice also that it occurs with Stannous fluoride and Stannous chloride equally. That shows you that the component of the Stannous fluoride, which is actually deactivating the LPS, is in fact the Stannous part of the molecule. This is important because not all fluoride toothpaste don't include Stannous fluoride. So Stannous fluoride toothpaste have this type of activity, not just a regular fluoride toothpaste. We did additional studies using a technique called mass spectroscopy, and we showed that the positive charge Stannous was able to actually bind to the phosphorylated region of the lipopolysaccharide, and this cartoon shows what that binding looks like, and that was the mechanism toward deactivating the LPS from reacting with the dye. And this, what you're seeing here, is the actual structure of the LPS that causes the inflammation reactions in your gums. So then we went on to reporter cell studies. So here, Stannous fluoride in vitro again is added to LPS or LTA from various sources, and the reactivity with reporter cells is determined. So this particular study was with using LPS, and we increased the concentration of Stannous fluoride, and you see a decrease in the actual reporter cell activity. This shows that stannous fluoride interferes with LPS and LTA activation at toll receptors. I should point out to you that not every antimicrobial that we study has this type of effect. So in the publications that I'll share with you at the end of our presentation tonight, you can go look, and some of those publications, for an example, would be a non-ionic antimicrobial like triclosan does not have any effect in deactivating the LPS or LTA from the bacteria. Instead, that type of agent may reduce the number of pathogens by decreasing their vitality or something, but it doesn't actually block their toxicity. So then we did an important experiment. We wanted to try to understand whether the stannous fluoride reactivity was occurring on the LPS or whether the reactivity was occurring on the actual reporter cells. And so we did a blocked experiment where we interacted stannous fluoride with the reporter cells, washed them off, and then interacted them with LPS. We used a special kind of endotoxin. It's a fluorescent variant. It's called Alexaflor. And when you take Alexaflor and you mix it with these reporter cells, they start to glow bright green. That's because of the interaction with the fluorescent variant of LPS with the cells. And when we added stannous fluoride, we were able to see that we blocked the actual interaction. When we added stannous fluoride to the reporter cells and washed it off, we still saw reactivity with the LPS. So where the stannous fluoride is deactivating the toxicity is directly on the bacterial LPS. Then we did an additional experiment to confirm that what we were seeing with the reporter cells, these special types of cells that were designed by Invivogen, that they were giving us the same answer that what we would see if we looked at cytokine production, actual activation of inflammatory processes in human cells. So we used human monocytes. Here we added LPS to the monocytes, and if you do that, you provoke all sorts of cytokine expression from the cell line. This is IFN, this is IL-1 beta, and TNF-alpha, so you can see they're all increased. We measured 8 cytokines in total. We saw the same effects for all of them. And as you increase stannous fluoride concentration, you in fact see a decrease in cytokine expression from these cell lines. So these data confirm the reporter cell findings, and they show that stannous fluoride reduces LPS provocation of inflammatory processes in the monocytes. So again, what we saw in the laboratory is our dye assays show that stannous fluoride binds to LPS and LTA toxins. The dye assay shows that stannous fluoride interferes with LPS interaction with reporter cells. The reporter cell assays show that stannous fluoride interferes with LPS and LTA activation of the actual toll receptors. And the human monocyte assay confirms that stannous fluoride interferes with cytokine production provoked by LPS. So these were very, very positive findings for stannous fluoride. And as I said earlier, not all antimicrobials show these effects. Some do, some don't. So stannous has been demonstrated to block antigen reactivity with toll receptors. And so here you see a cartoon showing that it blocks the interaction with these toll receptors. So the question is, do we see these effects in vivo? So we've run 4 clinical studies examining the effects of stannous fluoride, and in these studies we took the effort to actually measure subgingival plaque. This is very unusual for us in our business category. Usually we're doing all our measurements above the gum line. Most of the time they're doing measurements below the gum line. It's in academic circles. But in this particular case, we chose to do measurements below the gum line with subgingival plaque, and we also looked at metabolism and production of non-protein toxins in the saliva. And we've seen similar results in all of these studies. For brevity, tonight I was going to talk about one of these studies in some detail. So in this clinical study, we had a population which included 2 groups of people. One was a low gingivitis group, a low gum bleeding group. These people had less than 3 bleeding sites in their mouth, and they had pocket depths which were less than 2 millimeters pretty much. Then we had a high bleeding group that had more disease. They had greater than 20 bleeding sites. For reference, there are 168 sites in a mouth with full complement of teeth. So that's about 1/8 of the site's bleeding, so it's a pretty significant amount of bleeding. And they were allowed to have 3 pockets, which were up to 3-4 millimeters deep. These were our clinical populations. The product was used for 4 weeks. We copied the same design we did. It was an intervention design, but in this particular case, because we had so many samples, we just did a straight intervention. But it's the same design as we did in the control study, which I shared with you earlier, where we got a 70% reduction in gingivitis. So we screened them at baseline. We measured them clinically, and then we had them use the Crest Pro Health stabilized stannous fluoride toothpaste for 2 and 4 weeks, both the low bleeding group and the high bleeding group. The measures that we took at baseline, weeks 2 and week 4, included clinical exams of bleeding and gingivitis. We did some gingival plaque sampling, so we were very careful to remove all supragingival plaque, and then we took a curette and we went under the gum line and we sampled the plaque underneath the gum line for analysis using these new techniques we talked about. We normalized the plaques for biomass. And then we also took Metabolomics measures. We did NMR studies on the morning wake-up saliva samples. So we'll talk a little bit about each of these samples. So here are the clinical results. And as we've seen before, at 4 weeks, we had about a 55% reduction in gingival bleeding from the stabilized stannous fluoride toothpaste. And you'll recall that was that same similar percent reduction versus a positive control, or a negative control rather. So that's typical for what we see in these clinical designs. So this was fairly standard. The toothpaste was working as we usually would expect it to. So then we started looking at the actual measurements below the gum line. So these are endotoxin concentrations in the subgingival plaques that we analyzed by dye. And you can see that at week 4, we had a statistically significant, approximately 50% reduction in the endotoxin concentration in the subgingival plaque analyzed by the dye. Then we looked at reporter cell activation, and in this assay, we took subgingival plaques, and again, we cultured them with these TLR4 cells. And again, we saw a statistically significant reduction in TLR4 reporter cell activation in subgingival plaques sampled at 4 weeks after having used the stabilized stannous fluoride Crest Pro Health toothpaste. We used a different reporter cell line, a TLR2 cells, because it's a different reporter cell. TLR2 is activated by lipoteichoic acid, which comes from some gram-positive organisms. And we also saw deactivation of that reporter cell at 4 weeks duration with the subgingival plaque. And then lastly, we looked at cytokine expression. Now here we didn't study this in monocytes. We studied this in THP1 cells, which is another reporter type cell design. That was the cell that we used for the TLR2 incubation. But those cells actually will express cytokines as well, and in those cells we saw a statistically significant reduction in IL-8 production from those samples. So cytokine expression in the reporter cells was decreased as well. Then we also looked at the saliva analysis, and we saw statistically significant reductions in both butyric acid and propionic acid. For butyric acid, it actually started to measure upwards of 45% following 4 weeks use of the stabilized stannous fluoride toothpaste. And so these were very, very positive findings. And as I say, we've replicated these in a number of other clinical studies. We see the same thing each time. So from the clinical samples, what we've seen is that isolates from subgingival plaque showed reduced dye activity following stannous fluoride hexametaphosphate toothpaste treatment. The clinical samples from subgingival plaque showed reduced toll receptor activity for both TLR2 and TLR4 reporter cells. The clinical sample isolates showed reduced cytokine expression, and lastly, the salivary lavage showed reduced levels of the short-chain carboxylic acid toxins following the use of stannous fluoride toothpaste. Now, there were a couple of other things that we were able to look at in this study, and that's include comparison of the actual population groups. the low bleeding group and the high bleeding group to see how they differed in terms of their plaque toxicity. And then we were also able to look at the actual bleeding sites and non-bleeding sites, because those were sampled separately as well. So when we first look at the subgingival plaque from patients with high and low levels of gingivitis, we see that people that had high amounts of bleeding, and these are from the bleeding sites, but we saw that, you'll see in a second, that we see the same result at the non-bleeding sites. Their subgingival plaque showed higher toxicity than the plaque which was sampled from the low bleeding group. So the people who have more disease seem to have more toxic plaque. Now this isn't just, I need to say, this isn't just more plaque, this isn't just more dental plaque, because the supragingival plaque had been cleaned off, then the plaque is normalized for analysis. They actually have more toxic plaque below their gum line than the low bleeding cohort. And then this slide is rather busy, so I need to walk you through it a little bit. On the top set of bars is the data from the high bleeding group. The bottom set of bars is the data from the low bleeding group. The red bars are the bleeding sites that we analyzed, and the blue bars are the healthy sites that we analyzed. And what you can basically see is, number one, the high bleeding people had more toxic plaque, that is higher levels of this TLR luminescence. They had higher levels of toxic plaque at each one of the sampling times and so on than the low bleeding cohort. The second thing you see is that in the high bleeding group, the bleeding sites showed more toxicity than the non-bleeding sites. That wasn't true in the low bleeding group, so their plaques weren't as different. And then lastly, the utilization of the Stannous fluoride toothpaste was able to reduce both. And I would point out that in the high bleeding cohort, the levels of toxicity that we saw at the end of the study start to look more like what the baseline levels looked like in the low bleeding group. So that tells us what the Stannous fluoride toothpaste is doing in modulating the toxicity of the dental plaques as we use it. So what this says to us is that really the effect of the antimicrobial therapy, the Stannous fluoride therapy, in suppressing inflammation of the gingiva is really all about decreasing the toxic burden in the tissues, and it's facilitating the host management of the infection. So on the left-hand cartoon here, you can see that under high amounts of subgingival plaque with a lot of metabolic activity, and you have a lot of LPS and LTA, and all these toxins are activating the tissue to induce a significant amount of inflammation. And here you can see on the right-hand side the decrease of all of these metabolic. Now, we've measured LPS, LTA, and short-chain fatty acids. We've separately measured volatile sulfur compounds. We haven't measured enzymes and amides, I must say. But the idea would be to be able to decrease all of these things to reduce the signature of inflammation on the tissues and thereby reduce the amount of reactivity and bleeding. So we've done confirmatory studies, as I say, where we've looked in two other Pro Health Stannous fluoride formulations and seen the same kind of effects. And we also saw reductions in bacteria metabolites in a study of regimens where we included mechanical hygiene. These are some of the key publications. I can make these available to people if they're able to. I'm going to leave my email address, and I can certainly send copies of these to people. But these are the publications that we've had talking about this research. And what we really learned is that we were able to create some assays to measure toxicity. And in metabolic assays, we already knew Stannous fluoride was a very good inhibitor of bacterial metabolism, including sugar metabolism to acids, but we were able to prove that that includes the metabolism of short-chain fatty acids, which help contribute to gingivitis in the clinical situation. In the laboratory, we showed that Stannous fluoride binds to the endotoxin and the lipoteichoic acid from the periodontal pathogens. We showed that that binding deactivates the provocation of gene expression in the reporter cells. We showed that that binding deactivates the provocation of cytokine expression in the cells. In clinical intervention studies, we showed that this actually works, and in fact, it even works below the gum line. The subgingival plaque had lower dye reactivity, TLR reporter cell activity, and it had lower cytokine production activity when it was normalized and analyzed in our assays. And we also had lower levels of short-chain fatty acids in the salivary Metabolomics study. So in the clinical studies, we also saw that participants with elevated levels of disease had elevated toxicity, and this makes sense. In higher disease patients, the plaque from bleeding sites had more toxicity. This also makes some sense. In healthier patients, they showed lower levels of toxicity, and the bleeding and non-bleeding sites were similar. So the subgingival, interestingly enough, subgingival plaque from all patients at all sites was rendered less toxic with the use of a Stannous fluoride dentifrice treatment. Now, there are things, of course, that remain unknown. We still don't completely know why some people exhibit elevated responses to toxic plaque versus others. So we can't completely explain why some people have the higher levels of disease that you see versus the lower levels of disease. We've tried to look at their genotypes and so on, but we can't seem to see a simple pattern for that. We also don't know why some oral sites seem to evolve into chronic inflammation from the toxic plaque versus some other sites. So there can be two areas in the mouth that are right next to each other. One of them has gingival bleeding, one of them doesn't, and the plaque sample from the sites doesn't look so different. So we don't know what defines the threshold for health versus the chronic gingivitis, and we also aren't completely sure what the trigger is of more advanced periodontal diseases. But these are some of the things that our continuing research is looking at. So to summarize for you, antimicrobial Stannous fluoride toothpaste provides benefits in lowering toxicity of plaque, and it even occurs below the gum line. And I have to say, I started out in the 1980s. I did research on chlorhexidine, and we were doing sampling of microbial plaque above the gum line for the Food and Drug Administration, and we were showing them what we did to the plaque. And I remember the FDA was saying to us they were all very happy that we didn't see any changes in the microbial populations, and I was kind of surprised. I said, how does the chlorhexidine reduce gingivitis if it doesn't change the microbial populations? But we've done some studies with chlorhexidine on, chlorhexidine, it turns out, is a very good deactivator of toxicity as well, because it bonds to the phosphorylated sites on the LPS. So that kind of explains why we saw the efficacy we did. And the antimicrobial dentifrices, including those containing Stannous fluoride toothpaste, should be recommended, including Stannous fluoride, should be recommended for patients to provide optimal benefits with each toothbrushing. And the metaphor that I get back to what I started with at the beginning of tonight's webinar. I still, for the life of me, do not understand why professionals do not recommend antimicrobial toothpaste that have been clinically proven effective and endorsed by ADA and all sorts of people like that. Because the way I look at it is, you're going to be brushing your teeth anyway. You might as well be using something that decreases the toxicity in your mouth. And I look at it like airbags or anti-lock brakes. I mean, if you don't drive cars without anti-lock brakes or airbags anymore, and it's not because you know you're going to have an accident when you go out driving tonight. It's because if you have an accident, you have the protective feature designed into the car to help protect you. It seems to me that professionals should be recommending evidence-based, clinically proven products with maximum oral health benefits, and that includes antimicrobial toothpaste, it includes power toothbrushes, it includes dental flosses, and so on and so forth. So with that, I'm happy to address questions that folks in the group have tonight, and can't wait to have a dialogue. Can Stannous fluoride cause microbial resistance is one of the questions that was asked. Microbial resistance is typically not induced by these topical antimicrobials that we use. It's not the same type of effect as antibiotics. So we've tested Stannous fluoride toothpaste out to 3 years in cavity clinical studies and 2 years in periodontal studies, and we haven't seen any development of resistance of bacteria, nor have we seen a loss in the actual clinical efficacy of the Stannous fluoride over the time. So we haven't seen that. So this is the slide I wanted to show. Actually, what I'm more worried about than antimicrobial resistance is the development of patient susceptibility to periodontal disease over time where they don't have adequate hygiene. So this is a study that we carried out with Gosia Klukowska, and it was a study where we were looking at metabolomic changes, but we also looked at these clinical changes. And it was a very instructive study. So again, we had two groups of people. We had people with high bleeding and we had people with low bleeding. And in the high bleeding group, we measured them at baseline, we measured them clinically at baseline, and you could see big differences in gingivitis at baseline. Then we gave them a dental prophylaxis, and we gave them an electric toothbrush, and we gave them dental floss, and we gave them regular toothpaste, and we told them to brush like crazy. And during the period of time, they were actually able to reduce their gingivitis levels. So the high bleeding group was able to reduce their gingivitis levels all the way down less than 10 bleeding sites over the course of like a 7-day period. Then we had the people stop brushing their teeth and we put them in an experimental gingivitis protocol where they weren't allowed to clean their teeth anymore and the plaque was allowed to redevelop. And we were astonished to see how rapidly the high bleeding group actually redeveloped gingivitis. So the slopes of these lines, the redevelopment of gingivitis is nowhere near the same. So the high bleeding group is much more susceptible to getting more disease. We think the reason why this is because of epigenetic changes which occur in the actual tissues that make them more susceptible to chronic inflammation. So actually, when folks ask me if I'm nervous about microbial resistance, I'm actually a lot more nervous about people developing increased susceptibility to periodontal disease over time than I am microbial resistance. So that's my feeling about it. We actually have data to support that. Will there ever be a toothpaste developed with chlorhexidine in it? That's an excellent question. Chlorhexidine is very difficult to formulate in toothpaste because the concentration that you put an ingredient in toothpaste to be effective has to be about 10 times the concentration that you use in a mouth rinse. And so when we make a 1% chlorhexidine toothpaste, the amount of tooth stain that you get is just, we have very good stain control technologies now that we put in Stannous fluoride, and none of our Stannous fluoride toothpaste induce any significant levels of dental stain, but the chlorhexidine toothpaste are just terrible, so we haven't been able to make one without side effects, and they're also difficult to formulate. What are the side effects or drawbacks of Stannous fluoride? The main ones historically have been the difficulty to formulate a bioavailable form. So all of the original Stannous fluorides from the 1950s, they had bioavailable fluorine, but they didn't have any stannous. So none of the chemistry, none of the biology that you see tonight was taking place with those formulas because they just didn't have any bioavailable stannous. The more modern variants have bioavailable stannous and they're actually effective. The second drawback is obviously tooth stain. And when we first introduced bioavailable Stannous fluoride in 1995, we did not have that solved. But with the modern formulations, we have that solved. So now we run clinical studies for 6 months duration in dental office practices. And in those studies, we grade dental stain. We grade the amount of time it takes a hygienist to clean the teeth. And we have the dentist and the hygienist and the patient evaluate their teeth and tell us whether they can see stain. And unless the answer is 0 stain, the product doesn't get marketed. So yeah, the way to avoid staining is to use the Pro Health variants are actually quite good. Don't get confused about the naming of our products. You know, the mouth rinses we sell tend to cause more tooth stain than the toothpaste. The toothpaste we sell don't cause hardly any tooth stain. If you see some tooth stain in some patients, it can be easily removed with any of the whitening toothpaste. That's at least our experience. Inflammatory changes that won't go away in spite of all regular home care methods. Yes, that's exactly the thing that you worry about, is if someone has chronic, and there that requires a different invention. That requires something that works on the host susceptibility to the inflammation reaction, because I don't believe antimicrobials and mechanical hygiene, this is the type of effect you can produce. You can get them healthier, but it's very, very difficult to reduce their susceptibility. If they stop brushing or if they miss brushing, they're going to go right back to having inflammation. It's very, very difficult to convert them over to healthy, which is why professionals should be recommending to keep them healthy in the first place by using antimicrobial products. How large were the samples in the studies? How many? 20 people in each. Some of our studies, some of our later studies had upwards of 50, 60 people per group. So we've had significant number of people in our studies. Have you looked at the effect of stannous fluoride on superinfection of oral lesions? No, we haven't. We have some. We might have some practice-based studies where people have used stannous fluoride toothpaste, maybe the HD formula, but I don't know of any studies on superinfection of oral lesions. That's a good question. Something maybe we should do. What about the effects of xylitol? It's a very interesting question. Xylitol is an interesting type of, I don't know what to call it. And sometimes people call it an antibacterial, sometimes they call it a sweetener. Xylitol is really a prebiotic. And so what I mean by that is how xylitol works is the bacteria try to eat the xylitol, but they're not able to completely metabolize it for energy. So in the interior of the bacteria, it locks the bacteria up so that it can't metabolize sugar when you get exposed to sugar later. The problem is that mechanism works somewhat effectively when people are using chewing gums and lozenges and so on, because the xylitol gets into their dental plaque and it's able to produce their effect and so on. It's fairly straightforward. But when you put it in a toothpaste, the toothpaste contains surfactants, and the bacteria are not that active right after you use your toothpaste. They don't necessarily eat the xylitol. So the amount of xylitol that you have to use to produce any type of effect in toothpaste is upwards of like 10%, and then usually the effect is a very short period of time. And to my knowledge, the xylitol doesn't have any effects on toxicity of plaque. Do whitening toothpaste really whiten or just remove stains better? They remove stains better. And are they prone to create sensitivity? Yes, clean teeth hurt. So the more you clean, the cleaner you get the teeth, on average, some of these toothpaste can actually increase sensitivity somewhat. And some of the toothpaste produce a sensitivity response on their own because they're at very high ionic strength. Having said that, though, that effect is only experienced by a minor number of patients. Most patients that use whitening toothpaste, they complain that they might have some sensitivity, but doesn't stop them from using them. The amount of clinical benefit that you get out of the toothpaste isn't nearly the same as what you get out of our whitening strips, for example. So our tooth whitening strips are much more effective than any of the whitening toothpaste on the market, regardless of what they have in them. Do all stannous fluoride formulas perform similar efficacies? No, it depends on how you formulate them. Does formulation impact efficacy? Yes, in a big way. So that's why our formulas have all been qualified with multiple clinical gingivitis studies. You have to be very careful. The balancing of stain and gingivitis is extremely sensitive thing that you have to design for. What's the difference in stannous fluoride concentration in different toothpaste? They're all the same, 0.454% stannous fluoride. Some of them contain some additional stannous salts, which are added as stabilizers to increase the bioavailable stannous that's in them. Compared to regular toothpaste, how abrasive is the antimicrobial toothpaste? They vary. They're all well below the safety standard of 250. Most of our antimicrobial formulas are somewhere in the 150 range to help control the dental stain. These are really good questions, I must say. Can you briefly discuss the popular charcoal toothpaste whitening process? Yes, charcoal is really interesting because it's high surface area of charcoal. If you push that down in your gum line, one would think that would have a high affinity to absorb toxins and stuff below the gum line. So I would have a tendency to believe that has actually a fairly high chance of having some activity. I'm not so sure about dental stains, because I don't think the charcoal is hard enough to rub the stains off, and I also don't think that the stains are going to voluntarily leaf off your tooth onto the charcoal. So I don't necessarily think that mechanism works the greatest, but I am intrigued about people's use of charcoal because I have a feeling it might have more effect than we might think. Yes, the Stannous fluoride toothpaste are available in Australia. In fact, I spent a fair amount of time getting the permission from the authorities to place the label antimicrobial on our toothpaste. It's called OralB Pro Expert in Australia. For pediatric patients, this is a great question. So here is our dilemma with pediatric patients. Our Stannous fluoride toothpaste are labeled for multiple benefits, and that includes dental hypersensitivity. But dental hypersensitivity as a claim for the Food and Drug Administration is not an allowed claim for children because children don't have dental hypersensitivity. So because of the hypersensitivity labeling requirements, our products look like they're labeled for 12 and above. But to be perfectly honest with you, there's no safety reason why the Pro Health toothpaste can't be used in all. In fact, it's quite a good toothpaste for reducing acid production in the dental plaque in children, I'm sure. Based on your clinical experience, do you believe people with the inflammatory gene, that's IL-1, I'm sure, have effective results with regular fluoride toothpaste? Yeah, that's something we've checked for. We were thinking that perhaps the high disease susceptible group would all be IL-1, would be part of that IL-1 group, but that isn't what we saw. So they do see clinical benefits, but that doesn't explain their increased susceptibility to gingivitis. Now, I'm not going to comment on periodontitis because that's a separate issue. Okay, we already talked about the pediatric. Any changes? Are there any anticipated changes in the near future as to the composition of dentifrice products? Well, Lord knows they're always changing. I would dare say that in the future, I think we're eventually going to get a place where we start to add ingredients to the toothpaste that it's possible to treat these high disease patients. But that's someplace we're not quite there yet. That requires the use of some technologies which would affect their inflammatory susceptibility. Are products like ClinPro 5000? No, ClinPro 5000 part per million fluoride is the only thing 5,000 part per million fluoride is good for is for caries. It's a high fluoride concentration, but that doesn't have any indication for gingivitis or for any of the other indications that Pro Health has. But the 5,000 part per million is certainly highly recommended for people that have high caries susceptibility or some conditions which might lead you to need a higher dose fluoride. Are any studies done comparing hydrogen peroxide? Well, hydrogen, we sell a dual product at Crest HD that has Stannous fluoride in the first phase, and then it has hydrogen peroxide to help control dental stain and so on in the second phase. And that product's fantastic. I welcome you to try it. But hydrogen peroxide on its own doesn't have significant plaque and gingivitis benefits, which is why it was not recommended as a Category 1 ingredient by the Food and Drug Administration. Are there any other questions? Okay, if not, thank you very much for all of your attention. It was a pleasure being able to speak by webinar with you this evening. And if you have any questions, you have my email address, and so we can always send you copies of those papers and so on.